Thank you very much, Victor. Super nice talk, impressive. Um, Oh, yeah, you can take it. So as you might have seen, we have experienced some difficulties with the previous talk, but the difference is how we actually elegantly deal with it. So we keep wave propagating. And basically we are moving to the Michael's talk, which I think is the things that we are gonna prioritize. So what we are gonna do, we are gonna trim a little bit the hands-on session. So we are gonna make sure that we are actually finish on time for the opening ceremony, but we really want to benefit from, Michael present in the workshop, which is like a giant, has a lot of knowledge on the field. So with this being said, I leave the floor to Michael and take it away.
Okay. Thank you. Okay, this time I'm just going to bury straight through. So this talk is in 4 different parts. Some preliminaries going back to the sort of basic foundations and the empirical evidence for why we would take neural population approach. I'll be then speaking about whole brain neural mass models because a lot of people in the field are a little bit more familiar with those, and the gains and insights from those can be used to inform neural field models. So I'll speak through some of the theory of that, and then I'll be talking about some further applications along the line that Victor was speaking. So what I was saying, attempting to say last time, is that we start with a generative model, an unknown model, dynamical systems model of what's happening within the neural systems that we're interested in. We're not starting with the data and doing large language models or ICA or PCA. We're proposing that the data that we're observing is generated by an underlying physiological process. This picture is actually from Paul Nunez's 1977 book that I've overlaid. So what are the elements of that? Are the states, the X's, these are the neural states, firing rates, membrane potentials. We'll come to that. This is the change in those states. This is just a first order, dx to t, but we'll be looking at second order differentials in time and space. F is a function then to which we approximate all the physiology that we're interested in. These can be current-based or capacitance-based models typically. And then we have these inputs, which are coming from elsewhere in the brain. Or they can be noisy, they can be external stimuli, but these are external inputs into this little neural population that we're measuring. And we've got some control parameter. This is something that the brain might be self-organizing, like through neuromodulation. I read a lovely paper yesterday where people put in a probe into the medial septum and cooled. So it could be an experimental manipulation, or it could be a psychedelic manipulation. So F is a vector on these states. We're going to assume, in this case, 2-dimensional states. At each point in these state space, X and Y, membrane potential and firing rates, for example, we get this vector field. These then, according to dynamical systems theory, form unknown orbits which describe how the system evolves in time. And classically, these orbits flow onto invariant sets, which we call attractors. And in this case, you can see it's sort of a limit cycle. So this system's going to behave with some periodic oscillation. That's the sort of menu of dynamical systems theory. So this had a parallel history to neural field theory. Applying this approach to individual neurons was extremely successfully pioneered and more or less solved, and many of you in the room would know the Hodgkin-Huxley models. Hodgkin-Huxley model. This was a capacitance-based model derived from invasive patch clamp recordings of the giant cell axon. So I won't walk through these equations. They're first order in time. They involve opening and closing of membrane potentials and ions flowing in and out. And these can be some reduced and simplified, but they describe most of the types of chattering, bursting, spiking, dynamics that have been recorded at the single cell level. But I'm coming back to this in a moment. Most cognitive functions of interest to this field, perception, motor action, really don't arise from individual neurons. And moreover, these individual neurons are embedded in circuits, which are embedded in systems, which are embedded in networks. So they're very difficult to scale up from the single cell, scale up to the whole brain just through brute force. Individual spikes are very noisy as well, and behaviour on the whole is quite smooth, and I'll talk about how we get from these noisy, sparse spikes to the sort of smooth, large-scale dynamics that we observe. So it looks like we're going to need to move to a population model. And instead of the spikes encoding something, we're going to look at the mean firing rate of those spikes and maybe the variability around that mean. So this is what the enterprise looks like. Victor and I wrote an overview of this 21 years ago now. So suppose we have a reduced spiking model, and now we're going to integrate, say, 1,000 of them. This would be a brute force neural network simulation. But what we do is take time windows of these spikes. So these are 1,000 neurons. Each of these is an individual spike. They have underlying states. We're going to look at the distribution of those states. And if at some time we can approximate those states mathematically or just empirically with an easy probability distribution, this is kind of what we call a mean field approximation. And most of the time we can do this, but the trick is then to find an equation that takes the initial states and propagates that mean and variance forward in this particular occasion and captures the actual mean and variance. And so this is sort of the neural mean field approximation to neural dynamics. This is a more complicated equation that describes both the mean and the variance, but most of the time we're just looking at the mean. So does this make sense? And when we were doing this, it was a bit of a leap in the dark, but now of course you can get these beautiful whole field recordings. This is calcium imaging of the zebrafish larva published in 2024, and you can actually see from brains of small organisms the action potentials of many, many thousands of neurons. So in this particular paper I did with Brendan Munn and Max Shine and others, we took these single cell recordings and they're very sparse. So there's a mean firing rate, but there's very little variability. This is the sparse, noisy firing of individual neurons. And in this particular paper, Brandon then started doing iterative coarse graining. So taking pairs, and then pairs of the pairs, and then pairs of those pairs, and so on and so forth. Vic was talking about renormalization. This is a form of renormalization. And then at these higher dyadically downsampled scales, we similarly looked at the distribution of states. This can be done because we have 10 to the, I can't see over there, 15. It's a huge number of cells. And the main thing to take away from here is that these states get more and more Gaussian. So they get less and less kurtotic. They get more and more like a standard normal distribution. They become a Gaussian distribution just below the largest scale, a few scales down. And there's a lot more to this paper, by the way, that Brandon and Mac published last year in Cell. And also of relevance is that we had certain type of behaviours, wriggling larvae, or I should say swimming, and we had perceptual inputs to the risks of rodents. And it was exactly these meso and macroscopic population representations that were most tightly linked to behaviour. So please go and have a look at this sort of empirical validation of what we've been doing since 50s, according to John today. So a neural mass model, basically, these are the sort of multi-scale approximations that we can do. We can go from individual ion models of those to single cells, or their compartments, or just one cell. This is the sort of scale I'm looking at, and we can go for a scale up. So often when you present this, particularly to a hardcore biological neuroscience community, people get quite frustrated about all the detailed library of chemical synapses and neural cell types and where they synapse. Do we throw out of all of this? So this would be individual neural physiology. And we have many, many thousands of these. So using techniques, I've put the central limit theorem, but there's more sophisticated fluctuation approaches. These are incorporated into these population level models. So when you manipulate some of the individual cell synapses, you parametrically manipulate some of the population equations that we're putting into our models. So we're not losing the neurophysiology, we're just approximating over the many thousands of neurons within each population. This is a toy model that I did many years ago. I'm just going to use it to demonstrate something. It's pyramidal cells with potassium channel conductances and local inhibitory cells that get excited by the pyramidal cells and feed back slowly. So you get these spikes, and then you get these slow fluctuations in the local membrane potential, and this gives rise to bursting. These have got different time scales, and when they don't match quite integer, then you could get this form of chaos, which John blamed on 10 dark years, the medieval times of neural field modelling. So now what we can do is take one of these populations, put this equation in each population, and then connect them through the connectome. And this is something that I think many people in the field are now familiar with. And take our little toy models and put them in each population, and then connect them together and integrate them, hopefully do some mathematical formulation. There were some papers on this just under 20 years ago by a couple of us in the field. Now when you do this, each population has got, say, 2 or 3 or 4 dimensions. We've got maybe 500 populations now, so it's still 1500 dimensional, but it's a much smaller dimension than the whole brain. And these complex models actually collapse down onto lower dimensional manifolds, and this is what Victor was talking about, these manifolds that are much lower dimensional than the full phase space that organise the dynamics. And in this particular paper, we saw different types of waves, rotating waves, breathers, travelling waves. Applied mathematicians love these sorts of waves in different media, and we proposed a course of them at this whole-scale brain level. There's a lot of evidence in the field. Johanna's been working on brain waves for a while now. They're in just about every species, different types of recordings, different spatial and temporal scales. And maybe Johanna will talk about that a little bit later today. Now, where do these waves come from? Basically, if you look, and this is a compilation of hundreds of people's connectomics reconstructed from diffusion-weighted imaging. If you plot distance between nodes on the x-axis, and then you take log of some measure of the edge weight, you get this exponential, because this is log, so it's an approximately exponential distance function. This is what Victor was talking about. And this is the core organising principle beneath these neural mass models. So local neighbours are highly connected, and neighbours a bit further away are less strongly connected, and further away. And this is the synaptic kernel that pushes or causes the propagation of activity. And this is what is important for neural field theory. So now we're going to go to neural field theory. There's a lot of work, more and more, coming on neural mass models connected in this way. But we're going to stop in this neural mass madness, as some of people in the field might call it, and see what we can do at the next largest scale. So let's do an approximation at the macroscopic scale. Because that's what the simulations and that's what the brain shows. We don't see 1,500 dimensions of activity with a 1,500 dimensional model. We see maybe 5 or 6 dimensions. Okay, so this is what I was just speaking about, neural mass models connected on the connectome. It's almost an industry now. This is the underlying fibre length versus log weight. And now what we see here are intrahemispheric connections in black, interhemispheric connections in blue. And they're actually on the same underlying exponential distance function. And the red ones would be homologous connections from left to right cortex. So left motor to right motor, these are a little bit stronger than the background exponential distance rule. And you see a few little lumps here too. And I'm not going to go into those in too much detail here. So we can take the beautiful topological structure of the brain, and using an algorithm that James Roberts and I developed, we can resample the connectome that only preserves the exponential distance rule, but also the noisiness around that. So you see the red connections are now in this background noisy EDR. And when you do that, I mean, Peter and James Henderson had some foundational work on this. Just because these connectomes are spatially embedded, you actually get a lot of the underlying network topology. So you get modularity because of the lobus structure, you get clickishness, you get short path length. You certainly don't get everything, so there are additional topological constraints. But if you're coming to the convention centre from, say, Marseille, then the exponential distance rule will get you, maybe to a few hundred kilometres outside of Brisbane Airport. And then the topological arrangement has to do the rest and pushes these nodes apart and does obviously a lot of other things that give us our individual cognitive fingerprints. This isn't just in human connectome. This is the work that we showed. It's particularly consistent across subjects. You don't have to threshold. You can threshold by consistency to preserve the exponential distance rule. And it's also present in tracing studies from mouse and macaque. And John was talking about much earlier work on this exponential distance rule. So what are we going to do now? We're going to do, Victor was talking about integral forms. We're going to now look at our firing rate here. Q, I should go the other way, I guess. Yes, Q is a firing rate at some distant population. So this is now a graphical schema of what I showed before. The inputs are propagating through the cortex and through the white matter and yielding inputs at this other position R. And this can be written in integral form. It's a more general form than a differential equation, which I'll come to in a moment. It's just capturing this, that the firing rate phi is an integration over space and time of this connectivity kernel and the firing rate at Q. And all the time delays are accommodated in this integral form. This is probably the most important slide from my talk, that there is underlying connectivity in neural field models. And this is the more general form. I think this was the Nunez form. So what we're going to do now is put into W, this kernel, a complicated equation like this, but it's got the exponential distance rule. It's got these time delays. It allows for the spatial delays. It's got a normalization factor here. And into this equation, we put the exponential distance rule. So we're losing some of the spatial noise. And Victor was talking about the importance of those noisy anisotropic connections. But to first order, this is what we're doing. And then we do some magic in the Green's function. This is what John was talking about in the appendices with the Bessel functions. And you'll also I think you'll see it in the Yersa and Harkin model. When we do that, we get from that integral form to this damped wave equation. This is a differential form. But hidden in this equation is that exponential distance rule. So now we've gone from integral form, which physicists love, to a differential equation, which lazy mathematicians like myself love. Now, this second order spatial derivative is, this is from fMRI data actually. It's not the vectors themselves, it's the second differential, so it's the curvature of those vectors, which are in this case derived directly from bold data. So it's a second order differential equation. So isn't this lovely? We've gone from the Schrödinger equation of the atom 100 years ago to a damped wave equation for the human brain. And you can see this particle wave function actually extending because of the first order differential equation here, but I won't go into the details here. Now, this is not a closed set of equations. This is just the outward propagation. To close this system, what we're going to do is consider this phi, which I spoke about before, as incoming to our other point on the cortex. What happens when it's incoming is that it gets filtered through the synaptic dendritic complex. I've written it here as a second order differential equation, but it looks like this. The kernel of it looks like this. It then propagates. So we've gone from phi through the kernel to V, which is the local mean membrane potential. And then we go down to the soma and we get to this famous activation function where we've got V here and we've got Q. So we've gone from phi to V to Q, and then this propagates back outwards through the wave equation. So the wave equation is driven by Q. The Q turns into a phi that then changes the V through the synapses and dendrites, and then this goes to the cell somas and gets turned back into a firing rate. Victor's talking about time delays. Peter's team put in the thalamus with these characteristic time delays between the cortex and the thalamus, and that's also important. Okay, so some applications now. Here's our damped wave equation. I'm just going to, in the next 18 minutes and 21 seconds, look at some applications. Now we're going to ignore the spatial variability, but we've still got the whole neural field equations here. In particular, we also have these other two functions I spoke about, and we're going to have a look and see how that compares to neurophysiological data. And this is a figure from a paper that I did 20 years ago when I was Peter's postdoc. This is our neural field model. This is our some EEG data. But crucially, earlier work of Peter, 2004, on human brain mapping, showed that you don't just get time series that look like EEG data, but you actually get the whole spectrum. This shallow component here comes from the wave equation. This very steep component here, I think it's a product of Tularensis, comes from the synaptic dendritic filtering. And this peak here comes from the corticothalamic time delay, which is just as Victor showed, yields resonance frequencies and then can yield periodic oscillations if that loop is strong enough. Okay, so you can look at this. Victor was talking about instabilities. Instabilities in these systems come in a variety of different forms or plants or a taxonomy of bifurcations. This is a very simple Hopf bifurcation. As we increase one of these gains, there's a sudden bifurcation to an oscillation. When I was Peter's PhD postdoc, he said, look at these bifurcations, Michael, which I did. This 3 Hertz one mimics 3 Hertz petit mal seizures in quite fine detail because you've got the additional specific and reticular nuclei. You get these hooks that look like data. And just as you get spectra in the resting state, you get these harmonic structures in the model which mimic the harmonic structures in the data. This is a a petite male seizure, an absent seizure. Unlike what Victor was showing, these are global seizures of the cortex. So that's the 10 hertz one. We explored and found a 10 hertz oscillation, bifurcation. This is called a subcritical though, because you track up here and you suddenly burst onto a large amplitude oscillation, and to get back to the fixed point you then have to go backwards and fall off here. So subcritical Hopf bifurcations are also found mathematically. If you push the system from 0.85 say to 1, and then it jumps up to 2, and then you go back to 3, this is what the model looks like. Sudden large amplitude oscillations, and then back here. And this is a tonic-clonic seizure, the other type of generalized human seizure. This is what the data and the model looks like. And you get this harmonic frequency which suddenly blows out onto a limit cycle. And because you've come back down here, you've got this heavily overdamped post-ictal suppression of the EEG, which you don't get in the other seizure because it's symmetrical. Another paper I did on neural field theory, this time with Petra Ritter. We took resting state EEG data. We looked at the alpha rhythm, which is the canonical rhythm of the scalp EEG. When we took the amplitude squared, you get the power, and you see it's very bursty. This burstiness is present in just about every subject that you record with eyes closed. You can then look at the power, you can just bin it, and you can fit some Gaussian distributions, and you see that in just about all of us, we get these bimodal distributions. And you get trapped in the high energy mode, or you jump into the low energy mode. These are Poisson distributions, but you can see they're sort of stretched. So there's a bit of trapping going on here. So that's the human alpha rhythm. We took the neural field model again. We found a subcritical Hopf bifurcation with a much lower amplitude, and we drove it with noise. And you can see you get these bursty power fluctuations drawn from a bimodal distribution with this Weibull distribution, which is trapping. So this is, I think, a canonical model using neural field theory of the human alpha rhythm. What about spatial processes? So in the next 13 minutes, I'll talk about spatial processes. I was talking about the temporal dynamics of whole brain neural field models. So now we're going to put space back in and how we're going to do that. And I think maybe Richard might speak a little bit about this. Here's some inspiration. This is called a Chadley plate. You take a square plate or a plate with another geometric form, you sprinkle sand on it, and then you make it vibrate. When you make it vibrate, you get these pretty patterns. You can do it with a violin string. You can change the boundary conditions and you'll get different forms. Now the plate is obeying Newton's equations, F equals ma, adapted to a plate with a bit of stiffness. So it's obeying a wave equation. And these plates have different eigenmodes, different harmonic forms. And depending on what frequency you put in, you're exciting these different eigenmodes. And the sand goes to the nodal lines, not the peaks, but the nodal lines, these lines here that are not moving. And the peaks are in the middle of the sand patterns. So you actually, by doing this, you're exciting the geometric mathematical structure of the geometry. Are there vibrations on the brain? Now to achieve this, you can rewrite the wave equation in Fourier form. This is not my work, I'm just rehearsing work that was done. I believe Pedro made some contributions, Nunez and others, but formally this comes from Peter Robinson's work, which I think you'll see the citation here. You can then do a separation into spatial eigenmodes and their carrier frequencies. The eigenmodes can be solved or obtained by solving the so-called Helmholtz equation on the geometry of the cortex. And because of the beauty of computer science, you can now do these derive these solutions on very arbitrary curved two-dimensional surfaces. And so this is from Peter's paper in 2017. These are the fundamental and harmonic eigenmodes of the human cortex. The zeroth order, first order, second order. The higher the spatial frequency, the faster the temporal carrier frequency. And once again, this is done with spherical harmonics over 100 years ago. These are the solutions or the excited modes of the Schrödinger equation. So if you put energy into a hydrogen atom, this is where the electrons will most likely reside. And we can look at the same sort of eigenmode structure of the cortex. So we can do this. Traditionally, I've been coming to OHBM since about 2003. The very much dominant paradigm when I started to come, and it's still a very informative approach, is to take our fMRI data at each voxel and consider it to be scaled by some experimental manipulation plus some noise with the hemodynamic response function. And this is then thresholded in order to find these classic blobs. We're instead going to do an eigenmode analysis, and instead of having betas scale the amplitude of different voxels, I think I've got it here, we're going to have a coefficient that scales the amplitude of these different eigenfunctions, plus some noise. So it's the same sort of general linear model, but it's posed in eigenmode space rather than voxel space. And as you know, this was done by James Pang and Alex Finito a couple of years ago. Hundreds of data sets over a myriad of task sets were taken. The eigenmodes were obtained from the cortex, and then it was tested to see how quickly you could reconstruct the unthresholded fMRI data. So here's 100 eigenmodes, 10, 100, 200. And it's pretty visually clear that by about 100 eigenmodes, you've got a pretty good reconstruction of the raw unthresholded signal. And you can see that formally here, that you've reconstructed between with 0.8 to 0.9 accuracy from 100 eigenmodes, 100 basis functions out of 32,000. So this has a number of quite important implications. These eigenmodes from 200 down to 1 are all of spatial wavelength at least 6 centimeters. So most of the energy in the brain is at these lower spatial frequencies, not these tiny little clusters that you see when you threshold. This is consistent across subjects, it's consistent across tasks. So when you do a task, you're basically exciting different eigenmodes. We compared this to different types of modal representations you can obtain through the Laplace-Beltrami operator from the connectome, and you can just put in a simple exponential distance rule which is blind to the geometry. And in all of these different tasks, the geometric eigenmode outperformed these other geometric basis sets. An eigenmode can be obtained from a 5-minute T1 weighted scan. If you do a lot of pre-processing, the connectome eigenmodes can look very much like the geometric eigenmodes, but you've obtained a lot more data to get a geometric connectome eigenmode. So we formally in this paper compared the neural field equation to the neural mass model in terms of its functional connectivity for resting state. And the neural field equation with very few free parameters was more accurate than the complicated high-dimensional neural mass models. And this is coming from somebody, by the way, who basically made his early and mid-career research out of doing neural mass models. So I found these results extremely exciting. What you also see from the damped wave equation, if you ping the visual cortex, as the wave propagates forward, because it's a damped wave, it disperses and it gets slower. And this, in this particular paper, was also shown to recapitulate invasive neurophysiological recordings up the visual stream. Quite a famous set of papers to obtain this. Okay, so I think it performs pretty well. That's basically what I've been saying. Now we're going to go back to the whole spatio-temporal solution or framework. So we've looked at the temporal dynamics, we've looked at some of the spatial basis functions, but to get the full neural field equation, you need to go and do the whole thing. And this is just a prelude to Rich's talk, which will be this afternoon. Basically, you represent the damped wave equation with the eigenmodes, but you put in the two other equations to get the neural field. So just briefly, where's Richa? Yep, somewhere here. Okay. Rich's work shows that you basically, and this comes from some insights from James, propagate the system forward one time step on the eigenmode space, but then you pass the states through the dendritic filter and the neural activation function to reconstruct the full neural field, and then you do the modal decomposition and again and again. So there have been numerical simulation frameworks, NFSTIM and others, but this is much, much faster because for a 32,000 mesh, you're only integrating about 100 equations, not 32,000 equations. And very briefly, when you do that, you get something like this. So you get the peaks from the corticothalamic feedback, quite excited in this case, and you get the full spatial dynamics on the cortex, and coming out soon will be also on the hippocampus and on the cortex and hippocampus interacting. I think that's it. Thank you for persevering through some technical disruptions. Neural field work with Peter, Kevin and Richa, eigenmodes with James, Ben and Alex, numerics earlier with Stuart Heitman, now with Richa, whole brain neural mass models I did with James Roberts and Leonardo Goyo, and then this multi-scale modelling recently coming out in Cell with Brandon, Eli Mueller and Mac Shine, and they'll be here. So if you see Eli, Brandon or Mac, go up to them and say, Breaky showed me your beautiful work. Can you talk me through it? So thank you.